All right, thank you very much for inviting me for this session, and I'm really pleased to be with you guys for this session on the new horizon in the diagnosis of latent tuberculosis infection with Widas TB-IGRA. Before starting, I would like to set up my introduction on my end. I am Vaibhav GARUD, the Global Market Manager for immunoassays for bioMérieux, and I'm really pleased to be with you to share this information about our fully automated VIDAS TB-IGRA solution for tuberculosis infection detection. So moving to the agenda for our meeting today. So if you can move to the next slide, please. In this, the agenda that we will cover is to go quickly on the TBI burden and the diagnostic approach for tuberculosis. Then we'll discuss more about the breakthrough innovative TB-IGRA solution that we have with bioMérieux. And we'd also like to share the clinical performances of our Widas TB-IGRA when we compare it with QIAGEN quantiferon and other key, I would say, diagnostic assays on TB-IGRA testing. And finally, we will allow some time for some question and answers and for some discussion around this topic. So let's begin with the TB burden and diagnostic approach. So next slide, please. And maybe you can do one more click. So just as a quick reminder, we all know March 2024, March 24 is the World TB Day. And I think it's an important strategy by the WHO to really end TB. I think this day is celebrated as World TB Day. With that in mind, I would like to share tuberculosis, as we know, is caused by Mycobacterium tuberculosis pathogen. And TB remains one of the leading causes of death caused by this one single infectious agent. And again, this is as per the WHO report 2023. So tuberculosis is a major global health problem. And here you can see, as per the statistics, 1.3 million people die from TB each year. Again, around 10.6 million people fall ill from tuberculosis, and this is again alarming. And that also specifically from a pediatric children perspective, around 1.3 million children become ill and around 250,000 die due to TB. So definitely TB is a global health problem and one of the major challenges. And if you look into the next slide, the geographical scope, and you can see that definitely it's spread across the globe. Despite a lot of progressing knowledge on the disease, the new diagnosis tool, every day there are around 20,000 people who develop TB and more than 4,000 die. So definitely, if you look at and try to compare with COVID, so the numbers are very much close to COVID ones. But again, this disease has been for many, many, many years. So moving on to the next slide is more to give you a context about how TB, we can compare it with the COVID context. And if you see here again, in the TB incidence rate is estimated to have increased, by 3.9% between this pandemic area that we had between 2020 and 2022. Again, after 2 years of COVID-related disruptions, again, a lot of focus was more on the COVID, and that's why few people diagnosed or treated. Essential services were limited, or no focus more on the TB-related topic. But now, as we can see, after the merger global recovery that's happening in the number of people diagnosed with TB and treated, so this trend has started to reverse, or at least we can say it has been moderate now, looking at the damage impact of the pandemic. Maybe one more click, please. So here, definitely, now the impact of pandemic, the number of people dying or falling ill from TB is slightly, more stabilized. And now, if we go on to the next slide, where we will talk about the 2 stages of Mycobacterium tuberculosis infection. Again, TB is spread by inhaling of tiny droplets with an infected person who coughs or sneezes. And so this is a major spreading of the TB or active TB. So if you look at the first most important stage, which is the latent TB, again, here, with one more click. Yeah, in the latent TB, what's more important to understand is this is the inactive stage of the TB infection. Here, the patient are normal, they look normal, they are asymptomatic, they cannot transmit TB infection to others, but there is a high reservoir. They have a large reservoir for TB infection. And because if there is no systematic preventive treatment, if you're not able to identify those latent TB cases, there is a high chance, and maybe one more click, or we can just do the animation. There is a high chance that around 10 to 15% of this population from latent TB can convert to active TB. And just a quick glimpse on the active TB, and I think it's well known, active stage of TB infection where the bacteria is replicating, the clinical signs and symptoms are um, active and visible, or clinically diagnosis is possible here. And people with active TB definitely, need treatment. I mean, that's going to be a key prime importance for from a treatment perspective. So as we see the two different stages of disease, latent TB, what is important to remember is a huge human reservoir for TB infection, and around 10 to 15% of this population can be active TB in a couple of years or so down the line. So I think it's going to be very, very important that this latent tuberculosis patients population, which is the huge reservoir of the disease, needs to be diagnosed. And if you look at the next slide, what's the priority for WHO and TB strategy is to diagnose latent TB in order to prevent the progression of this to the active disease, and also to stop the spread of the disease. And this is where the WHO is really focusing more on to eradicate the disease by 2030. Now, when we say latent TB, and I think also Emily expressed that latent TB, 1/4 of the world population is latent tuberculosis, or can be infected with latent tuberculosis. So, who are the patients who need to be really at risk in terms of diagnosing latent TB? So, if you move on to the next slide, and maybe you can also do the animation so that we save time. So who should be really focused on the test for latent TB? As we cannot really test 1/4 of the world population. So focus will be on those risk group people. And here, as you can see on the top part of the slide, which says, people who are at high risk of progressing to active tuberculosis. And the group includes children which are less than 5, definitely immunocompromised individuals, persistent with HIV co-infection, the patients who are on immunosuppression therapy. These are the population who are at high risk groups. But again, there is a second group of population which is like a contact cases, or we can say that a high risk of contracting latent infection. But today they are low risk, or can be considered as also a moderate risk for progression to active TB. And in this, you can see the patients or population with above 5 years old, healthcare workers are the ones, homeless people, the ones infected with, I would say, injected with drug users, and also in some countries, it's the prisoners who are also being uh, 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 under the treatment, or I would say under the diagnosis algorithm. So yes, definitely there is a specific risk group that needs to be diagnosed for latent TB infection. Now let's look at the diagnostic pathway in the next slide, where we will see both latent and active TB infection. But our primary focus is going to be more discussion around latent TB. We saw that the TB infection exists in two different pathways, or I would say the two stages of the bacteria, one replicating and one being dormant. So in the active TB cases, as the bacteria is replicating, I think the diagnosis will really rely on the detection of the pathogen, and this is where we see smear microscopy be widely used, again, especially in the low income countries, despite of limited performances. But then again, the gold standard, which is the culture, which is also being used in a large scale. But again, it has its own drawbacks. It requires a long time because mycobacterium takes enough time in terms of the slower growth process. And then there are some more, I would say, modern, I would say not modern, but recent molecular biology techniques, which are also commercially available, that will help in detection of active TB cases. But our primary focus today, what we're going to talk about, is more on the latent TB part. And as late in the latent TBI part, you see the bacteria is not really accessible in terms of diagnosis, just like what we see in detection of pathogens with active. Here we use indirect methods. And when we say indirect method, it's based on the host response of the pathogen. So in the latent TB, one of the oldest techniques which we all have been familiar with is the tuberculin skin test, which is one of the oldest one, and is an in vivo test. And then there are the IGRA tests, which have been developed over, I would say now, more than a decade, and are in vitro assays. So we will now focus in the coming slide on the latent TB diagnostic approach for tuberculosis infection detection. So if you move on to the next slide, we again have to keep this in mind as for latent TB, there is no gold standard. The bacteria does not multiply, so it cannot be detected. Therefore, we all completely depend on the indirect approach, which is detecting the host response. So two types of tests that are used for latent TB, tuberculosis skin test, which is in vivo, and the IGRA assays, which are in vitro. Again, specific point, neither of these tests can really distinguish between latent and active, or I would say also say they are not able to detect even the progression of the disease. So it's very important to keep this in mind as well. You cannot differentiate between latent and active. They will predict the disease, latent tuberculosis, but again, you cannot distinguish between the two diseases. Again, these tests are reduced sensitivity when we talk about the immunocompromised individual. And in the next slide, I would like to just give you a quick summary of this existing test. For the tuberculosis skin test, as we know, it needs to be injected. You have to inject an antigen solution subcutaneously. The patient has to do a 2-time visit, once to have the antigen injected, and then finally to measure the outcome. So definitely it's slightly more in terms of turnaround time. You require 2 visits, and again there is variability also when you look at from a subjective who's going to read that measurement. So we also know there are a lot of publications available which talk about there is cross reaction between the BCG vaccination and the non-tuberculosis mycobacteria infection with related to TST stem. And when we talk about the IGRA test, which are based on the principle of measuring the immune cell response, where this in vitro diagnostic test is performed using the whole blood sample. It is being stimulated by some stimulation reagents, and I'll talk more about this in our principle of the assay. So when there is stimulation that happens, the specific tuberculosis antigen cells respond by producing interferon gamma, and this is being measured to detect the presence of latent tuberculosis infection. Now, as an organization, if you move on to the next slide, please, we as bioMérieux, and maybe you can take one more click. bioMérieux has been being a specialized leader in the field of in vitro diagnostics for more than 60 years. Again, we have a strong background in the field of infectious disease, and our fight is always we are always engaged to lead the fight against this infectious disease. And with that commitment in mind, we are here to really provide solutions, biomarkers that can really impact your clinical practice, thus improving the public health and developing solutions for today's major challenges such as tuberculosis. So if you look in the next slide, the detection, as I said earlier, the diagnosis of latent tuberculosis infection is key. The objective is really to prevent the spread of the disease and also to ensure right treatment for those patients and prevent TB transmission. So for the detection, and in the next slide, as I'm going to give more details about the test. Of course, for any laboratory, for any microbiology lab, performances and the reliability of the test will be a key criteria. But again, with Widas TB-IGRA, our aim and objective is also to ensure an ease of use of providing a fully automated solution without compromising today's lab workflow, I would say, and also which is also key for a lot of healthcare professionals. So with Widas TB-IGRA, we bring in this fully automation IGRA testing, and also it helps you remove all the current pain points which are there with the current IGRA test. Right from sample preparation, different steps that are involved, the number of collection tubes that are required, a lot of manual handling which is involved. And so we are trying to reduce all the pain points of current IGRA test with our fully automated solution, Widas TB-IGRA. And yes, with the Widas TB-IGRA, if you go on to the next slide, please. We are here to really answer your challenges, as I mentioned, to bring in that clinical value, ensuring better patient outcomes, but at the same time, improving your lab workflow, enhancing the laboratory management, and bringing efficiency to your laboratories. So now I would like to take you through a few more slides in terms of our concept, and thanks to our innovation and our R&D perspective, which the aim for development of this test was to really develop a functional assay, which will enhance the clinical value, but also bring an ease of use and ensuring your laboratory management to the easiest and the best possible way in the laboratory. So let's move to the next slide, please. And the system that you see here is the VIDAS 3. So the Widas TB-IGRA assay is available on this platform. Again, it's a fully automated TB-IGRA solution. All the steps, including the incubation, the preparation of stimulation, I would say stimulation steps, the reading, everything is taken care by the instrument. So all the steps that are required for performing an IGRA test are managed by this equipment. So I would like to give you a short glimpse of a video which will talk more about how the test or the key highlights are featured on the Widas TB-IGRA platform. So can you please play this video? Thank you. Thanks, and maybe we can move to the next slide. So as you have seen, the Widas TB-IGRA is truly an innovative concept. As first you saw, one single patient tube is used, which means there is no pre-analytical step, no internal incubation, no centrifugation. Second, again, all steps of this assay are fully automatized. When I say that, it is the whole blood pipetting, the incubation of the stimulation phase, and also the measurement of this interferon gamma release that has been released. And again, definitely, as I said, with this concept, we allow standardization of all critical steps, which are patented again. And Widas TB-IGRA demonstrates also strong clinical performance, which I will be taking in the later part of the presentation. Now, let me go into the theoretical principle of the IGRA test. So again, there will be some animation. So science concept, as I said, this is based on the capture of antigen specific of pathogen by cells, which we call it as antigen presenting cells, like macrophages or dendrite cells. These cells process the antigen, okay, and present they're present on the surface, which are really bound to those histocompatible complex. So if the subject or the patient has been previously exposed to this kind of an antigen, the memory T cells will recognize this antigen complex and lead to activation of these T cells, the CD4 and CD8, which will produce this interferon gamma release. And this is what, one more click maybe, please. Yeah. So yeah, so once this interferon gamma is released, this is what is being measured by the system. So the interferon gamma will provide information regarding the potential presence of the immunodefense against the specific pathogen due to prior contact, or as I said, contact cases with TB patients. And now let's look at how does it happen inside our system. So the different steps. So as I said, it's an in vitro stimulation of the patient sample that takes place in three different steps. One, maybe you can just do the animations. Correct. Yeah. Okay. Perfect. So the first step, as I said, is the nil, the stimulation reagent on the top, which acts as a negative control. So which allows the measurement of basal level of the interferon gamma in the patient sample before the stimulation. Then comes the mitogen, which is like a positive control to check whether the patient T cell functional does its function. So the lectin molecule that is contained in this reagent triggers a non-specific T cell stimulation. And then finally, the specific antigen stimulation reaction, which happens, which is called as antigen AG, where the antigen contained in this reagent triggers T cell stimulation, showing evidence that the previous contact with the pathogen. So this is how the principle really works inside the system. And now there are different reagents which are also used to mimic this kind of reaction. And if you move on to the next slide, so for Widas TB-IGRA assay, the one that we have, our stimulation reagents, especially the antigen reagent, it includes a specific peptide, which is mentioned, I think, ESAT and CPEP, which is specific Mycobacterium tuberculosis antigen. And these peptides are very carefully designed and formulated by our internal R&D teams. And we know that know-how of the scientific information that is available about CD4 and CD8 cell, because they are a major interest in the TB immune response pathogenesis. And as expected, we can clearly demonstrate, and it's been characterized by also some flow cytometry studies where the antigen reagent of Widas TB-IGRA. And again, just to come back on the competition, if you look at quantiferon, which has 4 tubes, and the fourth tube which does the wonder for this CD4 CD8 response, whereas in Vidas TB-IGRA only one single whole blood tube does all the work that the 4 tubes are doing in the quantiferon assay. So with our studies, and we have a publication that has been published in November 2021 by Delia Goletto, who is an Italian renowned KOL. She has clearly demonstrated and characterized with the help of flow cytometry that the antigen present reagent of Widas TB-IGRA induces both CD4 and CD8 T cell response, again with a cohort of subject which includes active TB and latent TB patients. So the antigen that is used in the Widas TB-IGRA is clearly designed and formulated to ensure maximum sensitivity and specificity for the assay. So with this design, post-design information that I shared with you on the Widas TB-IGRA assay, now let's look at the clinical performance of our test. And maybe you can move to the next slide, please. So again, while performing the clinical study for Widas TB-IGRA, we have carefully, ensured the clinical performances were evaluated again across the world with different sites. So there are 19 sites which were used during the study. The clinical performance or the characteristics of TB-IGRA were evaluated almost around 1660 patients or subjects, and with 3 different populations. Again, this is very, very important to explain. And if you go to the next slide, please. The study populations that were selected for our clinical performances to monitor the sensitivity, the specificity, and the agreement, positive and negative agreement of the test. So patients with active TB, were one set of population where we really focused on the sensitivity part. Then we had a mixed risk population for MTB infection. And then there is also a population which is at low level of TB risk, where we can say that the blood donors, or let's say even the low endemic countries, like for example in France. So this quantiferon TB Gold Plus assay was used as a comparative assay. So all the study was done with this comparative platform as well. And if you look at the performances of our sensitivity, and if you go to the next slide, please. Here, as you can see, for the number of patients that were studied with Widas TB-IGRA, we represented a very high sensitivity. Again, these are the patients which are active TB cases, which were confirmed by culture positive, active TB cases. And here you can see with Widas TB-IGRA, our performances in terms of sensitivity was high. Again, one important factor to make a note here is the level of indeterminants. And here you can see against quantiferon plus, our indeterminant that was observed was only 2. So very low indeterminants. That's one key advantage with Widas TB-IGRA. Again, thanks to the automation, thanks to the stimulation, development reagents that have been developed by our R&D, which really ensures a stronger performance with the Widas TB-IGRA assay. Now, on the next slide, we'll look at the specificity. For the specificity study, as I mentioned earlier, the blood donor population was selected. And again, with Widas TB-IGRA, we were able to have a specificity of 97.6%, which is good, and I would say comparative with the comparative method, which was quantiferon gold plus. So this was the data that was presented, and which is also part of our pack insert claim for Widas TB-IGRA in terms of the specificity. And then talking about the test concordance, the negative agreement and the positive agreement. So on the next slide, as you see, we have a very strong concordance when we look at the agreement between Widas performance versus quantiferon, and very strong, above 90% agreement for both NPA and PPA that was observed for the Widas TB-IGRA with mixed risk population. And on the performance side, the last slide I would really want to highlight is also about the percentage of indeterminants, because this is important to also focus here, because Widas TB-IGRA had fewer indeterminants. And if you can go to the next slide, please. Yeah. So with Widas TB-IGRA, we had very few indeterminants. So our indeterminate rate was only 0.1%, whereas with quantiferon plus, it was 1.2%. And there have been publications which show a very higher indeterminate rate with quantiferon plus. So overall, with the clinical study that was performed for Widas TB-IGRA, we had a very good sensitivity, specificity, and a strong concordance with in terms of the positive and negative agreement, and very few indeterminates as compared to the comparative analysis. And last, I would like to conclude by saying, maybe this next slide, please. Yeah. Okay. For the data and the performances that I have shared on the Widas TB-IGRA assay, which we have launched again in March 2024 this year. So if you go to the last slide, please. Sorry. Yeah, thank you. So with Widas TB-IGRA, we are really proud to say that this is the first fully automated solution today for the diagnosis of TB infection. Again, as we saw earlier in the video as well, all the steps are included as a part of the principle or the process of this this assay. And we saw from the performances, we had a very good sensitivity, specificity, and agreement with the competitor. So again, Widas TB-IGRA can really bring in a big, I would say, change management in terms of the workflow, because it's a fully automation process. Less than 16 hours, you're able to deliver a test result. So that's the beauty of Widas 3. Less manual intervention, chances of errors are minimized because of the automation steps. So no manual intervention during the stimulation steps, because they are very crucial. Any contamination can lead to a batch failure on the other platform. So this is where I would like to say that with Widas TB-IGRA, we are here to really provide a reliable solution for the IGRA testing in your laboratories and to answer the needs for the clinicians' decision making. Thank you very much. I would say that's all on my end, but I'm happy to take any questions that you may have regarding Widas TB-IGRA. Over to you.
Yes, thank you. I would like to thank you very much for the nice presentation. I would like to see if there are questions. I don't see in the chat box if there are. Let's check. We have a lot of participants. I don't see. Tishani, if you can. Let's wait for a moment because we put that they can make their comments or make the questions. The participants can make the comment or type their questions in the chat box. Just wait for a moment to see if we can get the questions. Maybe Tishani can help me with that.
There's no question yet
There's
in
no
the chat,
question.
nor the Q&A box. But
Oh,
we have one.
okay, we have one question. The first question is if it is an FDA approved diagnostics system.
Yeah. Thank you for that question. So today we are GMAD or CE marked assay. We are in the process of submission to a US FDA. So again, with US FDA, there is a separate clinical study that is required for the US population, which is ongoing at on our end. And again, the timeline intervals will be slightly longer than any other regulation. But yes, we are in the process of US FDA submission and approval. So we keep you posted once this is done. But today it's GMAD and C marked assay.
Okay, thank you for this answer. We have another question. It said, I'm not sure if I miss it, but can you say how long does the test take from sample preparation to get results?
Okay, very interesting question, and I'm sorry I could not really. I didn't want to go into the technical part of the system, so I've not details showed that. But yeah, I think it's a important point. So from sample collection, as I said, you need only one tube which needs to be with a whole blood, 4 ml of tube, which needs to be collected. So from sample collection, let's say, to the performance of the test, including everything like incubation, performance of stimulation step, the system takes a total time of around 16 hours. So let's say within 17 hours, you are able to report an IGRA test on the Widas TB platform. However, if you look at other players, there is a 24-hour incubation plus the different steps that are involved. So a larger time window. So you can still save at least minimum 4 hours or maximum it can be more hours on the Widas 3 platform.
Thank you. I want to make sure because we asked the attendees to put the question in the chat, but I have to I have to reconsider. They can put it in the Q&A chat box, so then we can read it out.
Yeah.
I have a question. Is in your experience how it rolled out in the countries that it is all introduced, where is this test mostly used? In public health settings? And I have to explain a little bit. In the Caribbean, we don't in all the island have in the hospital the public health setting. So what is your experience? Is it a clinician setting or more outside in a public health laboratory that is not in hospital? What will you recommend?
I think I would say it's a mix depending upon the geographies. But as you mentioned about the geography where that's more of public setting in your case, we had a similar example in countries like Romania, Serbia, and so on, where there is a lot of public hospitals, institutes which do this kind of testing. And so we are present today with more than 40 countries on this test. And I can say there's a proportion of this test being done in both the settings, in public and the private settings. So in France, again, it's private, I would say, in most of the laboratories. In countries like India, Indonesia, it's a mix of both India, public and private. In the European market, again, it's largely hospital settings where the test is being performed. And again, the test is also performed for like in-house, I would say, screening for some in some countries. In some countries, it is used to monitor those or immunosuppressant treatments. So for patients with rheumatoid arthritis and all those cases, also the test is being used earlier. So there's a mixed, geographies. It will depend on varying upon the geographies, but yeah, it's been widely used in both the settings, public and the private settings.
And since you mentioned the clients you can use it for, what is more used for the immunocompressive patients, and what is the experience in your country, how it is used, and all the other countries you refer to?
Yeah. So as I said, I mean, for the feedback that I have from markets like Serbia or Romania, or even in countries like more in the European context, let's say, a lot of these tests are performed for ensuring the therapy, right, for immunosuppressed individuals. And the reason why they have switched to Widas TB-IGRA is the one advantage that they found was about low indeterminates, which was a pain point for them with different methodologies they were using earlier. So I think with Widas TB-IGRA, as you see, and we not only show in our clinical performance, we see a less indeterminate results. So the need for repeat testing after a couple of months is really minimized. So you have a very good concordance in terms of reporting when we say with Widas TB-IGRA. So yes, I think it's positive feedback that we are getting about our test in different markets today. I would say we will be happy to share some clinical cases and success stories in the later part of this year. I would say definitely, for sure.
Thank you. We have another question. What is the average cost of the equipment, and what is the maximum and minimum sample that can be processed at a time? I think the first one.
I will only take the second question, which is the sample volume, because I'm not a commercial guy and I don't have the rights to say. I will let the team explain that later on. But for the sample volume, as I said, a patient volume of 4 ml is required in a tube. That's the only requirement, 4 ml tube.
I think this question is more processed in one time in the system, not
Yes.
the volume, how many tests can be performed in the system.
Okay, so how many tests can be performed in the system? Okay, so with Widas 3, thank you. Sorry for that misjudgment. So as I said, Widas 3, this test can only be performed the Widas 3. So as you see, there are 3 strips that are required. One for the antigen, positive control, and the negative nil control. So for one patient sample, you require to put in 3 strips. So with Widas 3, you're able to perform 4 tests at the same time. So with one equipment, you're able to perform 4 tests at the same time. So let's say a workload of around 20-24 tests can really be fulfilled. I'm talking about a week's workload, can be easily fulfilled on this automation platform. So one system is able to cater to workload of 20-24 tests per week. And so if you calculate it annually, let's say 1,200 to 1,500 tests a year.
Okay.
Hope that answers the question. Yeah.
I think that is for sure. And we have another questions. Question, it is, how come the VIDAS TB-IGRA has better sensitivity on active TB disease?
Yeah, again, as I said, the population that was used for measuring sensitivity for active were all positive patients for culture. And again, as I mentioned, thanks to the stimulation reagents that are being used, again, the antigen that we have is very specific, and it's been definitely something from an R&D innovative concept that we have, which really ensures this sensitivity. and we have conducted several studies to ensure this clinical performances. So later, this year we will be presenting in the Union Congress, in the coming Union Congress, the performance along with the poster for our performances. So that's let me be on a marketing side. I would say it's something to do with an R&D innovative concept that has led to this high performances from a sensitivity part, and thanks to the antigen that's being used.
Another panelist asked, is it a continuous loading system on VIDAS 3?
Yes, but as I said, the capacity is for only 4 samples. So for TB-IGRA I'm talking about. For other tests, definitely it is. But for TB-IGRA, you can only have 4 samples. You can have 1 sample at a time, or you can. So it depends upon the section which is empty, right? So as I said, if the 4 slots are booked, you will not be able to do. You'll have to wait for the final test results to come out. But if you're performing only 1 test and there are 2 patients which come later on, there are three positions still vacant. So I cannot call it as a fully open loading system, but yeah, up to 4, you have the flexibility to run 1 sample or 4 samples at the same time.
Another question of the attendees: what makes the indeterminate rate lower with the VIDAS TB-IGRA?
Yeah, I think the most important reason, or I would cite, and what we have observed is, and again the feedback from the customer is, making this test full automation is very unique thing, because the most important challenge is preparation of, let's say, stimulation reagents, or doing that steps where there is a high chance of contamination. With Widas TB-IGRA, everything happens inside the system. So you have only one tube that you load the strips and the spurs that we which is the Widas concept, and then you just place the stimulation vials which are provided by in the box on the system. So right from pipetting to mixing of the reagents, and everything happens automated. The incubation also happens inside the system, and I think this is one of the reasons which really leads to a far better outcome when we look at our results. And this is what has been cited by a lot of our KOLs who are working on this clinical performances of Widas TB-IGRA. So I think thanks to automation, which really brings in or tries to prevent this pre-analytical errors that we see in most of the cases.
Let's see if we have more questions. I don't see more questions. Let me go to the chat box to check. No. Okay. No. I make sure we don't. Oh, yes, we have one more question. Okay. This question is, have you experienced labs introducing the VIDAS TB-IGRA while maintaining their current workflow? Can the VIDAS TB-IGRA be used for screening purposes for patients suspected of being infected? You get it? You get the question?
Can you please repeat the first part of the question?
Okay,
I was not
yeah.
very
Okay, I thought so. Have you experienced labs introducing the VIDAS TB-IGRA while maintaining their current workflow? And then, can the VIDAS TB-IGRA be used for screening purposes for patients suspected being infected?
So again, on the screening part, I would say it's quite complex to give an answer on that, because again, it will depend upon different contexts. So I would say I don't have an answer today for that second part of the question. But to answer your question on the workflow management, and I think this is an important advantage of using Widas TB-IGRA. And we had a recent publication from the French study, a study that was conducted at St. Joseph Hospital in Lyon, France, where we have seen the total time to result, as well as the number of steps and the handling time that is required, which brings in a lot of workflow efficiency with VIDAS TB-IGRA. So it was again done because the customer was using the quantiferon plus assay earlier. And with VIDAS TB-IGRA, they have really seen a drastic change in the, not only from the reporting perspective, the time to report, but also from a workflow management. And maybe I'll be happy to share that the poster that we have for this workflow study management. But again, we would appreciate if there is any such opportunities at your laboratories, hospitals. We would be happy to work with you in that on that prospect. But yeah, there is one publication which is today available, or one data which is available today for the workflow management.
Okay, we will make sure if we have the presentation and also the paper that we will send it to our participants.
Absolutely, that's okay with us. Absolutely,
okay.
Sure.
I have one more question. If there is an error during testing, will the system report it directly or until finalization?
Yes, if there is any error, let's say a mechanical error in the system with fluidics and other things, or maybe something to do with the test strip or the reagent that you have added to the system, there will be an automated alarm which will highlight in the beginning. If it's something to do with the processing, it will highlight immediately, and in case something you missed to put an reagent, you will have based on the nil values and everything, the background values, we can clearly recognize what's gone wrong with the process. But again, the chances of error will be only due to some mechanical issues in the system. But again, VIDAS is known for being sturdy and reliable. But again, that 1% chances are always there. Yeah, if there is something wrong in the performance of the system, it will be highlighted with an alarm.
Okay, thank you. Let's see if we have more question, or this was all the question of the participants for today. Let I switch to the chat box. No, I don't see more questions. First, I want to make sure I announce to the participants that they can fill in the evaluation form and get the certification for participating to this webinar. I have put the link in the chat box. Please check it out and fill it in. And the rest me no more than thank you, Mr. Garud, for so much insightful presentation. Thank you for sharing your expertise with us. And I think this will help us and make us aware of a new diagnosis tool for latent tuberculosis in the Caribbean. Thank you very much. On behalf of Farminpex ISRA Lab, I want to thank you for the presentation. Once again, this presentation is supported and powered by bioMérieux. Thank you everyone for participating to this webinar, and have a good day. Thank you so much.
Thank you, everyone. Have a good day.
Thank you.